Hello, my name is Kobe Christensen, and this is my journal club presentation. So for some quick background, I chose to do a paper that was involving the immune system in zebrafish and how it affects regeneration of stem cells in the zebrafish and how that can be used in human therapies. I chose the zebrafish because I'm currently doing research in Dr. Sandquist's lab regarding zebrafish, and I just feel like they're interesting, so decided to choose an immunology paper that tied human stem cells to zebrafish stem cells. So, for starters, zebrafish are unique because, unlike mammals, zebrafish have an ability to regenerate their central nervous system. So they can regenerate their brains, spinal cords, the retina in their eye with their own stem cells. And unlike in mammals, in mammals, if you have a spinal cord injury, then you'll remain paralyzed. In a zebrafish, if you have a spinal cord injury, you might be paralyzed for a few days, and then the spinal cord can regenerate and they can swim again. So, a keystone of regeneration in zebrafish and stem cell therapies in humans is the immune response. So, researchers are trying to understand how the immune response affects regeneration in zebrafish, hoping that that can give insight into how the immune response affects stem cell transplants in humans. So I chose a paper that focused on inflammation as a critical factor for successful regeneration of the adult zebrafish retina in response to diffuse light lesion. And kind of the goal of this paper, again, is if we can discuss how the zebrafish retina regenerates and how stem cells in the zebrafish respond to the immune system and inflammation responses, we can kind of figure out how stem cells might respond in human retinal transplants. So, some background on the actual study itself. For context, this figure here is not actually in the study. This is just to kind of get an idea of what we're talking about when I'm mentioning some of the cells that are in the zebrafish. So this is just a diagram here of the human retina. So in human neurons, inflammation can lead to permanent damage to photoreceptors in the retina. And in the zebrafish, what this study aimed to find was, does inflammation in the retina damage the stem cell response, or does it actually necessary for the stem cell response to occur? So they hypothesized that with previous data that they have where they know that the immune system is important and inflammation is important in the proliferation of stem cells in the zebrafish retina, they decided to go ahead and immunosuppress the zebrafish to see how the immunosuppression would affect the stem cell response. And they also thought, well, if we know that immunosuppression will probably decrease the stem cell response, do you think we can induce a greater stem cell response by inducing inflammation? So this is what they decided to go ahead and take a look at. So in the first experiment, they performed a light lesion. So they shined a really bright light on the zebrafish eye, which would kill the photoreceptors in the retina. And they used a transgenic fish where leukocytes, red blood cells, specifically microglia, would glow red. And as we've learned, microglia are kind of like the phagocytes of the central nervous system. And photoreceptors here are dyed with green fluorescent protein. So under the microscope, they show green. So what we can see here is that this is the control retina. And what we see here is you have these microglia that are kind of hanging out in the peripheral areas of the retina. And we have all of these photoreceptors here. You can see that they're intact. And obviously, since this is the control retina, the light lesion has not been performed yet. And here you can see we have a pretty good arrangement of these rods and cones. And here, the microglia, they appear as kind of like these red, stringy cells, which is kind of how microglia are depicted if you look at a diagram of them. And then here, so 2-DPL, DPL stands for days post-lesion, so this is after they shine the really bright light. What you can see is that there are a very much decreased amount of these green cells. So these photoreceptor cells have been killed, and what you have is these red microglia that were, before they were in more of the peripheral areas of the retina. They have kind of migrated to the areas where a lot of these red cells are dying. So what this indicates to the researchers is that these microglia must be doing something where all of the photoreceptor cells have died. So we know that there is an immune response involving microglia, that they're doing something where all of these photoreceptors have died. And something else interesting that they noticed, so here in the control fish, you see that these microglia, they have like a long stringy appearance. And here in the injured fish, they have this kind of bloated appearance. So they thought that that was interesting, that there was a phenotypical change in the microglia themselves, that they had changed shape. So in figure B, what we're looking at is we have L-plastin is a leukocyte cell marker that is used to visualize white blood cells in the retina. So here, this ONL stands for the outer nuclear layer. This is closest to where the photoreceptor cells are. So out here are the actual photoreceptor cells themselves. And what we're seeing here is that in the control fish, there are no white blood cells out in this outer nuclear layer, because there hasn't been any damage to the photoreceptor cells. Here, 2 days post-lesion, you can see all of these cells that are marked with this L-plastin cell marker, these leukocyte cell markers. You have all of these white blood cells that have now migrated to the outer nuclear layer. So we know that they're doing something in the outer nuclear layer to promote regeneration. And we'll further investigate as the study goes on. And then here in C, this is just a kind of quantitative version of this model over here. So in C, what we see is a numerical representation of the amount of white blood cells that are seen after the lesion is induced. So here we can see before the lesion is induced, and then an increasing number where at 2 days post-lesion, there's the maximum amount of white blood cells that are located in the outer nuclear layer that are probably repairing or doing something to the photoreceptors, and then kind of a decrease in those white blood cells as the photoreceptors are repaired. And then in figure D, what is interesting is, so here we've been talking about microglia. In figure D, we're now talking about neutrophils. So neutrophils are dyed blue with DAPI. And as you can see in the control retina, there are no blue neutrophils down here. So neutrophils have not yet migrated into the retinal space. But 12 hours post-lesion, you can see that there's a whole bunch of damage. So here, this extracellular space here is completely black. You can see here in the control fish, you have kind of this white shadow-looking extracellular matrix. This is, normal compounds that you'd expect to see there. Here, it's completely black. So the photolesion has wiped out anything that's in there. What's interesting is here you have this appearance of all of these neutrophils, and all of this white stuff here is the nets that we learned about in our last exam. So the neutrophils are casting out their nets and doing things to repair the extracellular space here. So researchers understand, okay, so there is an immune response that involves microglia, that involves these neutrophils, and they're migrating to the spaces where these photoreceptors have died. So okay, so now we kind of have an understanding, the researchers had an understanding that there was some type of migration to where injured cells were. So what they decided to do from this point was to give the fish a potent immunosuppressant called dexamethasone, and the point of this was to see, can regeneration take place in the absence of the immune response? So in figure A, we have just the timeline of the treatment here. So 10 days before the lesion, before the bright light was shown onto the fish, we have this dexamethasone, a powerful corticosteroid that is administered to decrease the immune system in these fish. So what we see is in figure 2A here, this is the timeline. In figure 2B, we see how dexamethasone has decreased the amount of leukocytes in the retina. So L-plastin, again, remember that is a leukocyte cell marker. So here in the dexamethasone fish, you see there's a whole lot less of these red cells here, which indicate the white blood cells. And in the control fish, so these control fish that were not given any immunosuppression drugs, you see there's a lot greater response of these white blood cells after the injury. Okay, so with less white blood cells, which is expected, right? Because the researchers had given an immunosuppressant. What they wanted to find out next and before I explain that, here we have L-plastin. Again, this is just a quantitative analysis of this image here. So you can see that the control fish, which is marked MEOH for the methanol that they were placed in, you can see that the white blood cells, they increase and then they decrease, right? They're indicating this is the point where they're kind of cleaning up the area, helping the retina to regenerate, and then they decrease as the retina regenerates back to normal levels. Here in the immunosuppressed fish, these never change, right? The white blood cells never really increase. So figure 2D, here we have a marker that is called PCNA. So PCNA is a proliferating cell marker, and basically what it indicates is in the zebrafish retina, you have these cells, these stem cells, that as they proliferate, as they divide, and as they grow, they give off this PCNA, which is marked here. So in the control zebrafish that just had the lesion and no immunosuppressants, you see all of these blue cells, right? And all of this white up here, which is indicating that these Mueller glia, which are the cells that give way to the stem cells, so they're kind of like the parents of the stem cells, these Mueller glia are able to regenerate, and you have all of these regenerating proliferating cells that are showing up in the retina, which are going to help to restore the retina and restore vision in the fish. But in the immunosuppressed fish, what was really interesting here is we see there is a lot less proliferation of those retinal progenitor cells. So the researchers had to ask themselves, why is it that we understand that there would be less white blood cells, but why are the white blood cells initiating a response here to regenerate the retina? So here, again, we see that this is just a quantitative analysis of this photo. Here we see that there's a lot less regenerating cells in the dexamethasone fish than the control fish, which obviously leads to poorer regeneration of the retina itself. So here in F, 2F, this figure shows the proliferating Mueller glia. So now these Mueller glia are stained green, and as you can see, these Mueller glia are kind of these long stringy cells. And in the control, you have all of these long stringy cells that span all the way across, and all of these white progenitor cells. And in the dexamethasone fish, the immune suppressed fish, you see there are a lot less of those proliferating cells. So kind of just, again, confirming what we know, that for some reason, when we decrease the immune system, we're having a decrease in amount of stem cells that are actually going to the area and fixing the retina. And again, here's just a quantitative analysis of that, where we can see that in the control fish, which is this light gray here, you have an increased amount of proliferating cells, versus in the immunosuppressed fish, we have not as many proliferating cells, so less stem cells in the retina. So 2H here, one of the factors that we know from prior research in zebrafish is that there is a protein that is called MMP9. And MMP9, essentially what it does is it goes around the extracellular matrix and it cleans up any debris, any dead cells, and it kind of makes room, if you will, for new stem cells to take place and turn into rods and cones and restore vision. So we know that MMP9 has this function, and we also know that NF-kappa B, which we've learned about in class, this is a precursor. This is an inflammatory cytokine that is a precursor to this MMP9. So the researchers decided, okay, well, if there's a decreased immune response, is it possible that we have a decreased amount of NF-kappa B, and potentially that is the reason, because there's less NF-kappa B, which results in less MMP9. Maybe the extracellular matrix here is so messed up with a whole bunch of stuff that it's unable to allow the progenitor cells the progenitor stem cells to take place and regrow the retina. So they tested this by marking NF-kappa green with this green fluorescent protein. So NF-kappa beta is green here, and you can see that you have all of this expression of this protein. And here in the immunosuppressed fish, you don't really have much expression at all. And then the MMP9, this is an array that is showing the MMP9. You see all of this MMP9 activity here in the internuclear layer that is doing work to clean up the extracellular matrix where a whole bunch of dead photoreceptors are. And here in the immunosuppressed fish, you really don't have much, if any, of this MMP9 doing that work. So researchers thought, okay, well, it's possible that through further research we find out that the reason why the inflammatory response is so important is because we need MMP9 to clear up the extracellular space so that we can allow for retinal progenitor cells to grow. So then they decided in experiment 3, they said, okay, well, if we know that decreasing the immune response decreases the amount of stem cells that can reach their final destination, then is it plausible that if we increase the immune response without damaging the tissue, if we increase the immune response, can we increase the amount of stem cells and the amount of healing that goes on in the retina? So what they decided to do is they take an undamaged retina and they inject it with 3 different compounds. So we have 2 of those compounds are immunogenic compounds called flagellin, and then a toll-like receptor, which we learned about earlier in the semester, called zymosin. And they knew that these 2 things would upregulate the immune response, and the 3rd compound that they injected into the retina was just a control. So they knew that it wouldn't do anything. It was just a control to make sure that they weren't killing cells as they were actually injecting substance into the retina. So the actual process of the needle entering the retina, they wanted to make sure that wasn't killing too many cells. So they're injecting it with a control substance that they know will not kill any photoreceptor cells. So here in figure A, they just have a timeline of when they performed the injection and when they did the analysis. So that's here. And DPI stands for days post-injection. So 2 days post-injection, they took pictures. 4 days post-injection, they took pictures. So here in 3B, we have this tunnel plus marker. And tunnel plus is essentially a cell marker that indicates cell death. So they can go ahead and measure how much of this tunnel plus protein is present. And if there's a lot of it, they know that there's a whole bunch of cell death that is happening. If there's not very much of it, they know that there is not cell death that's happening. And here they decided that this is not a statistically significant difference between the control and then flagellin and zymosin. These are the 2 immunogenic compounds. There was not enough of a difference here for a statistically significant difference to suggest that any of these compounds were causing actual tissue damage. So there's no cell death in the retina actually occurring here, which is important, as we'll describe later on in this figure here. So L-plastin, again, this is the leukocyte positive cell marker. And what this is trying to show is that between just the control injection versus the 2 immunogenic compounds, there wasn't a marked increase in white blood cells. And the point of that was to show, we don't want to damage the tissue. We don't want to increase the actual white blood cells that are present in the tissue, but we want to see if we can increase, without introducing white blood cells to the area, if we can increase the amount of cell proliferation that's happening in the retina without causing damage or white blood cells to actually physically go to the area. The thought is, in humans, is it possible that we could inject immunogenic compounds along with the stem cell therapies that we inject to increase the outcome of the stem cells taking in the human retina? So here in D, figure 3D, again, it's this PCNA, which is that proliferation of stem cells. It's showing the cells that are dividing. In the control experiment, we have no cells that are dividing. This is expected because the tissue was not damaged, so there would be no regeneration that needs to occur. However, in the flagellin and zymosin injections, what they realized is there is a whole lot of cell proliferation going on here, which indicates that stem cells are migrating to areas where there's no injury and proliferating just because they injected these immunogenic compounds. So that's important to know because we don't actually need to damage the retina to introduce stem cells. And figure 3E is just showing the same data, but here with the numbers. So here it's showing the increased stem cells, and the control, where there was no increase in the stem cells. Figure 3F here. So this is showing they decided, they had a theory about this NF kappa B and what it does for inducing MMP and how that's important for helping stem cells take. So here in the control, NF kappa B is marked green. You see there isn't really a whole bunch of it here. So this is the control. You wouldn't expect there to be much because there's no injury. But in the flagellin and zymosine injections, you have this increased amount of this NF kappa B, which indicates that we should have, because NF kappa B is a precursor to MMP9, more MMP9 in both of these areas. And here, that's what this figure G shows, is you have more MMP9. Again, MMP9 is responsible for going around, cleaning up the extracellular matrix, and making it easier for these retinal stem cells to take and adhere in their environment so that they can turn into, someday, rods and cones and repair vision in the zebrafish. So this was interesting to them, and they also decided that they were going to take a look at HER 4.1, which is a part of the Notch signaling pathway, which is generally upregulated when there is retinal cell injury. So they know that this protein exists when there is retinal cell injury, and what the goal of this was to show that even though they did not induce tissue damage, which is shown here by the control, there's no HER 4.1 because there's no tissue damage. When injecting flagellin and zymosine, there's this increase in HER 4.1. So with them knowing that there's an increase in HER 4.1, there's an increase in MMP9, this suggests that there's an increase in retinal progenitor cells, which is seen here. So by injecting these compounds, we can actually force the retina to regenerate even when there is no tissue that is damaged, which is really interesting because, again, if we can understand how these mechanisms work in zebrafish, we might be able to induce them in humans, which, as of right now, because we're mammals, we can't regenerate our retinas. But with stem cell transplants and potentially the induction of immunogenic compounds like these, we might be able to increase outcomes of stem cell transplants. So the conclusion that they came to was that an acute inflammatory response, they hypothesized that if they were to immunosuppress a fish, it would damage the inflammatory response, and thus you wouldn't get as good of a retinal response, and where these stem cells are regenerating the retina. And their study supports that. So they noticed that the stem cell response was inhibited when they had an immunosuppressed fish, and they also, hypothesized that maybe if we ramp up the immune system, we might be able to ramp up regeneration, which is what we saw in that last figure, and they proved that to be accurate as well. So again, this study, provides insights for investigating potential benefits of the acute immune response in humans who are receiving stem cell transplants, and how potentially modulating their immune system could lead to better outcomes for stem cell proliferation and migration when these people are getting stem cell transplants, which maybe one day could help with the restoration of vision in humans.